Hi, everybody, and welcome back. Today, we're going to be looking at monohybrid crosses. We're going to go through how to lay one out, how to work out a Punnett square, and then we'll work through one or two examples that you'll commonly see. Before we do that, I just want to cover all the terminology first so that you know how it all fits into the whole Punnett square and how to work it out. So, a monohybrid cross is mono, meaning one, is that you're going to cross one trait at a time. In other words, you're going to cross color or height, and you're going to keep it as individual genes. So when we speak about mono, we mean one cross. And we have genes and we have alleles. Now, a gene codes for a trait. It's a section of DNA. But you can have variations of that trait, and that's what we call an allele. It's a variation. In other words, you can have the eye gene color, and then alleles of that would be brown, blue, and green. And so they're variations of the eye color. The next important thing about monohybrid crosses is that we use letters to represent our alleles. And often a capital letter represents dominant and a small letter represents recessive. Now, the lettering I've chosen here is simply as an example, but if you want to express a dominant trait, you would use a capital T. If you want a recessive trait, you would need to express it as a small or lower case t. I'm going to explain exactly when and how you use these as well in the Punnett square, and also how Mendel's laws dictate how we use these letters. Now, technically, you can have combinations of these letters, and they will produce the different geno and phenotypes. So when we refer to a genotype, we are referring to the combination of those alleles. Two capital T's, a big letter and a small letter t, or two lowercase or small t's. But those letters represent a physical or outward appearance that we may be able to see. And so we call that the phenotype, and that is the physical appearance. Now, I haven't given these letters any meaning. In other words, I haven't related them to any physical trait. But I will use an example now as how you can actually view these correctly and what they actually mean, whether it's the genotype or the phenotype. The final part of our monohybrid cross and the terminology associated with it is something called homozygous and heterozygous. Homozygous is the way in which we write the genotype, and it tells us the collection of letters. If something is homozygous, it can be two capital T's or it can be two small lowercase t's. Now, they will also tell you in the question that it may be homozygous dominant, which means that's two capital letters, or if it's homozygous recessive, it'll be two lowercase letters. Heterozygous, on the other hand, means different. So that means that hetero will have one capital letter and one small letter. It's important to know that we only use these words and terminology when we are referring to the genotype. We don't use these to describe the phenotype. Now, the next important component of our monohybrid crosses is to look at Mendel's laws. And these are really important because they make sure we understand how to read the Punnett square, but also how to construct it correctly. So we do need to know these off by heart. And the first one is the law of dominance. And in a cross where organisms are purebred, which that means that the alleles they carry, for example, must either be two capital T's or they will be two lower T's. That means they're purebred, they have no other alleles to offer. In that contrasting, in other words, short and tall, black and white, only the dominant trait will appear. Recessive alleles will always be masked by the dominant allele. And so that means that you only need one capital letter T in order for it to be visible in the phenotype. In other words, if you ever have one capital T and one lowercase t, the dominant trait, which is the capital T, will always be visible in the phenotype. You'll be able to see it. The only way to see the recessive trait is if you have two lowercase t's. That then brings me now to the law of segregation. Now, the law of segregation states that characteristics are controlled by pairs of alleles, which we just saw earlier. Every time I've drawn my alleles, I've written them in pairs. And what happens is that during meiosis, they will separate into different gametes. Therefore, only one allele from each pair will be present in a gamete. In other words, if I was constructing a gamete from the following alleles, I had a capital T and a lowercase t. When they go into their respective gametes, they will go separately from one another. So one gamete will have a capital T, and the other gamete will end up with a lowercase t. So, how do we actually lay out a genetic cross and how do we do a Punnett square? So, the first thing that we always need to do is we need to start off with the P1 generation. The P1 generation refers to our parent generation. And what you're always going to do is you need to write their physical characteristics first. Now, you would take that information from the paragraph that they would have provided with you, and perhaps you will say a black mouse cross a white mouse. You then need to write out their genotypes. Now, remember, the genotypes are referring to the lettering. So let's stay with black and white, and I'm just going to use this as an example. But if I had a pure black mouse, it'd be two capital B's, and a pure white mouse, I would use two small letter b's. And somewhere in the paragraph, they would have either told you that white is recessive and black is dominant, or they would tell you what the children are and you can work backwards. But I'll show you how to do that in the next example. Once you've written out those characteristics, you then need to formulate the gametes. So in the column to the left-hand side, you would write gamete, and right below the parental genotype, you will write the word meiosis. Then what you will need to do is you will need to separate your gametes. So you're going to place one capital B a little bit of a distance away from the next. You can also use commas in between your letters. I like to use circles around mine only because it makes it really easy for the marker to know that these individual alleles are now separate from one another. They're in actual cells, they're no longer attached to one another, and just make sure that nobody misunderstands what I've written. After that, you are going to then have to include the word fertilization. And that is where you are now going to go into your Punnett square. And so before you start doing your Punnett square, in the side column, you are going to write F1. F1 refers to the filial generation. In other words, the outcome of the children. Now what we need to do is we need to insert our gametes into our Punnett square. Now we learned also that there's the law of dominance, but there's also the law of segregation, meaning that when we put in our alleles from our parents, we need to make sure that we keep the pair of alleles that came from one parent in the same column. However, they're separated by these lines, and that's the law of segregation. The other column going down is going to have the other parent's alleles. Now what you need to do is you need to substitute the values in. And it's very similar to maths in that you would use like a FOIL rule. You would then multiply this letter B with this letter, and you would then write the answer in here, which would be capital B, lowercase b. And then you would do it again for the far letter B, and you would do the same thing, and you would basically multiply the letter in. Now that you complete the entire Punnett square, and you must do it for the second set of letters as well, you now will need to write the pheno and genotype of the resultant offspring. And so what we always like to include is the genotype. Now the genotype is going to have to either be homozygous or heterozygous, and you need to include the lettering with that. Now in this example, there are only heterozygous examples. So you would write big letter B, small letter b, but then you also need to include the amount. Now you can decide, you can have 4 out of 4, you could have 100%. It just depends on the question and whether or not they want you to write a fraction or whether they want you to write a percentage. The final piece of your Punnett square is the phenotype or the physical outcome. Now, I said to you in the beginning that I did a cross on a black mouse and a white mouse. Now, if black is dominant and it is represented by this capital B that we wrote up here earlier, and if each one of my resultant offspring in my Punnett square have at least one, that then means that all the individuals in this cross will be 100% black. And you will need to write that down under your phenotype. Now, there aren't any ratios in this example. For example, there aren't 3 black to 1 white mouse, but I'm going to show you how that also looks in the next example. So let's look at this very basic example, and it will just reiterate what I was explaining in the previous Punnett square example. So let's say you get this question, and this question says, in a genetic diagram, cross a true breeding yellow pod with a true breeding green pod. Now, when they refer to true breeding, what they mean is this. They mean that the alleles must be the same. In other words, there must be two capital letters or two lowercase letters. But let's read the rest of the information so we know what we need to do. It then says that green is the dominant color. Now, when selecting a letter to use, often we use the capital letter from the dominant trait. Now, in this example, green is the dominant color, and therefore we're going to use the letter G. Now, if green is the dominant color, then my true breeding green pod should be two capital Gs because it's true breeding, it's pure. My yellow pod, on the other hand, is what would be termed the recessive trait. And we now know that the recessive trait can only be represented by two smaller lowercase g's. Now, if we cross them, what would they look like in their Punnett square? Now, of course, if we follow the whole outline of the Punnett square diagram, there are a few things that are missing in my picture, but the most important part I want to show you is the cross in the Punnett square itself. And you can see here quite nicely as well with the pictures as a visual. And you will notice that when we take our true bred green pod with our true bred yellow pod, there are our two recessive alleles for the yellow and two dominant for the green. And when you multiply it into the Punnett square, you will notice they all come out as being heterozygous. And all these green pods are a mixture of the two parents. Now, this is important because what it does is it shows you that every time you mix one purebred or true bred individual with another, you will always get 100% the dominant trait in terms of the phenotype. But what happens if you take two of these individuals and you cross them with each other? In other words, I might take this individual and I do another cross with them. So now here's my second example. What happens if we were to cross two heterozygous flowers with each other? Now, I'm going to use a different example now with flower color. I'm not going to use pods this time, just so you can visually see the difference. And so it says we're going to take two heterozygous flowers, cross them with each other, and it also tells us that purple is the dominant color. Now, because purple is the dominant color, we are going to have to make sure that purple is represented by a capital letter. Now, in this cross, they've chosen to use the letter B. I know that I said that you can also use the letter P, but this example, they've used B. And essentially what it means is that these two flowers are a big letter and a small letter. So if I make it match the diagram I'm going to put up now, that means that these flowers will be a capital B and a small b. And we're going to cross them with each other. Now, this is what it looks like. If we were to take the two purple flowers, because they will be purple, why? Because they both have a capital B in their alleles, and we do a cross with them, we get a slightly different combination in the first cross. You will notice that we have three individuals that have a capital B in them, and then one that has none. And what you're seeing here is the ratio that heterozygous crosses create. In other words, whenever you cross a heterozygous organism with another heterozygous organism, you always get a ratio of 3:1. In other words, you will get three organisms that will have at least one capital letter, like the three purple in this picture, and the one refers to the white recessive individual over here. Now, the final component of this cross is that we must make sure we write the phenotype and genotype out correctly. Now, the phenotype I represented just now by this ratio of 3:1, and they may ask you for the ratio in the question. But if you have to just simply write out the phenotype, 75% will be purple and 25% will be white. But it's important that that just doesn't translate automatically into what the genotype is. There is 75% purple. However, not all the purple flowers have the same genotypes. 25% of them are homozygous dominant, which represents, if we look back at the diagram, this individual over here. Then we have 50% that is heterozygous, and that then represents these individuals which have a mixture of our two lowercase and uppercase letters. And lastly, we have a homozygous recessive individual, 25%, and that is this white flower. So you'll notice that the geno and phenotype percentages are not always the same, and it's important not to just copy the phenotype percentages straight down. So let's quickly look at some terminology recap. We learned about a monohybrid. A monohybrid is when we cross one trait at a time. We either do color only, height only. And within those monohybrids, we have alleles and genes. Gene refers to the code, and that means that we are looking for a code for a characteristic. And alleles are variations of genes. In other words, you can have the eye color gene, and the variation of those eye colors makes the alleles. We then looked at two very important laws, the law of dominance and the law of segregation. And we also looked at the fact that organisms that have a dominant allele present will physically express it. In other words, that's when we see organisms that have capital letters in their genotypes. And so, whenever there is at least one capital letter, the dominant allele will show in the phenotype. In recessive organisms, on the other hand, they need two lowercase letters in order to be seen. And that refers also back to the law of dominance, in that in order to be able to see a recessive structure, you need to have two recessive alleles. We then looked at homozygous and heterozygous, which refers to the way in which you write the alleles, homo meaning two of the same, hetero meaning two different letters. We looked at how to use a Punnett square to work out a genetic cross. And finally, we spoke about pure or true breeding individuals. I'll remind you that a purebred or a true breeding individual is an individual that either has two capital letters or two lowercase letters. And often they have contrasting characteristics like tall and short, white and black. Thanks everybody, and I hope this video helped you, and I'll see you again soon. Bye!